Welcome to this third embryology lecture series on development and congenital malformations of the central nervous system. In this particular third lecture of the same, we are going to focus on development of the brain as well as that of the ventricular system of the brain. Maybe just to orient you on where we are, I chose to divide the development and congenital malformations of the central nervous system lecture into 4 parts for the sake of easy delivery. So in the first part, which we've already covered, we looked at neurulation and neural tube disorders. In the second part, we looked at development of the spinal cord, and at the same time, we looked at spinal dystrophic disorders. So those two we've already covered, and the lecture recordings are as shown. If you wouldn't listen to the two, I'd advise you that maybe first you listen to those two, because the story we're going to do right now largely builds on those first two, and especially the first one. Well, in this particular third lecture, we are now looking at development of the brain and that of the ventricular system. There'll be still another last lecture, which will be focusing on on the congenital malformations of the central nervous system. But because we've covered the ones associated with the spinal cord and spinal dystrophic disorders, this one will be largely focusing on those ones of the brain. Right, so this is our third lecture. This is where we are. This is what you're going to learn in this particular lecture. So I'd wish to first name the phases of development of the human central nervous system, and just bring to your attention that the human central nervous system develops in 4 phases. We'll be highlighting those 4 phases, and we'll be stating the key event that occur in each phase. That will take a bulk of this particular lecture. Another thing that we'll remind ourselves is about the neural tube. We'll remind ourselves about the parts and derivatives of the neural tube. Then something new in this lecture, we'll look at the brain vesicles. There are 2 types of brain vesicles, primary and secondary brain vesicles. We'll name them, and importantly, we'll also look at the derivatives of these vesicles. So that's our agenda for this lecture. Much of the story will be on the first agenda. That will be a bit recurrent in the whole lecture, as you're going to see. Let's start with just highlighting that first agenda. What are the phases of development of the human central nervous system? As I've already mentioned to you, the human central nervous system develops in 4 phases. We have the phase of dorsal induction, which involves formation of the neural tube. Once that has happened, then we have the phase of ventral induction, which involves induction of the primary neural tube so that it can form vesicles that give rise to the brain, what we call the primary and secondary brain vesicles. The phase of neuronal proliferation and migration is the phase where now neurons migrate from deeper zones of the neurotube to superficial zones of the neurotube with the aim of developing the cerebral cortex. It is at this phase that also we have gyrification and sulcation appearing. And then we have the phase of neuronal myelination, which is the last phase, where now axons of neurons are covered with the myelin sheath. So those are the 4 phases of development of the central nervous system. Now, just a note is that these 4 phases are not necessarily that one must end before another begins. As you're going to see, some of the phases actually do overlap. But in terms of events, these are the well-defined events that occur during CNS development. So as we discuss development of the brain, we'll be discussing development of the brain based on these 4 phases. Now we can start therefore with the first phase, which is the phase of dorsal induction. The phase of dorsal induction is the phase where neurulation occurs. So this is the process of neurulation. We've already mentioned something about neurulation in the first lecture, and so you're familiar with it, but just a few things that I want you to carry forward in this particular lecture. There's something called the notochord. We saw it in the first lecture. And we saw that the notochord induces the overlying ectoderm to thicken, then the ectoderm undergoes folding in the process that we call primary neurulation. So notochordal induction causes primary neurulation. Primary neurulation occurs between the 3rd to the 4th week of development, and it leads to the formation of the primary neurotube as well as neural crest. But primary neurulation does not take care of the whole neurotube. We are aware from that lecture that the most caudal part of the neurotube is not formed from primary neurulation, but is formed from secondary neurulation process. And secondary neurulation process is neurulation by mesenchymal condensation and then cavitation. If you remember, there's a process there that we talked about, which is called mesenchymal epithelial transition. The cells in the caudal part undergo mesenchymal epithelial transition, and then there is condensation and cavitation, leading to formation of the secondary neurotube. While the first neurotube, the primary neurotube, forms from the 3rd to the 4th week, secondary neurulation is never really completed until the 2nd month of development. That's unique, really. So I want you to capture that the times are not really that one must end before another begin. There's some overlap, as we're going to see. The second phase of ventral induction will have already started way before this 8th week of development. So there's some overlap, but there's some information also I want you to capture, because ventral induction will be largely focusing on the primary neurotube and not the secondary neurotube, and so there's no big deal. Anyway, so the take-home message is that dorsal induction involves neurulation process. Now that dorsal induction involves neurulation process, which leads to the formation of the neurotube, let's remind ourselves about the parts and derivatives of the neurotube. So I want you to remember that story, that neurulation is a process of formation of the neurotube, which is the embryonic primordium of the central nervous system, and that we have 2 neurulation processes, primary neurulation and secondary neurulation. And that primary neurulation is the one that is induced by the notochord. Secondary neurulation is not induced by notochord. So if you understand that, then I can show you the primary neurotube. This is the primary neurotube. The first image shows you neurulation, where now we see that the primary neurotube is closing, and from the lecture you remember that the closure begins from somewhere in the cervical region and then occur in a zipper manner, both cranially as well as caudally. With that understanding, we then realize that there'll be some opening cranially and opening caudally, what we call the cranial-neuropause. The cranial-neuropause, what we call the neuropause. The cranial-neuropause, which is this one, closes about on day 24 to 25 there, and the caudal neuropore, which is this one, closes about 2 to 3 days later. So usually the cranial neuropore closes earlier than the caudal neuropore. That was primary neurotube, which is represented by this particular image there. And then on the caudal end, we see something happening. We can see some condensation there, representing the beginning of formation of the secondary neurotube. In the second image, we see that whatever has condensed is now undergoing cavitation. So this is basically the secondary neurotube, and this is a primary neurotube. Usually, the primary and the secondary neurotube join. And so we have one big composite neurotube. By the time this is happening, we are now maybe on the 8th week of development. But all in all, we now have a neurotube. Remember, the primary neurotube is up and the secondary neurotube is in the most caudal region. So this is the final neurotube that has just been shown to you in the previous slide. So this is neurotube from primary neurulation. We see it, it is responsible for forming the cranial part. This is a secondary neurotube from secondary neurulation. It is responsible for forming the most caudal part of the neurotube. So that composite neurotube is a neurotube that has formed from both primary neurulation and secondary neurulation. This story happens in mammals, birds, and reptiles, and because you are mammal there, so this story also happens in you. Now, in terms of the derivatives of the neurotube gives rise to the central nervous system. But how? The cranial 2/3 of the neurotube give rise to the brain. The caudal 1/3 of the neurotube give rise to the spinal cord. Don't be tempted to say that because the spinal cord is long, so perhaps it takes the lion's share. That's not true. The truth is that we are looking at the mass of tissue. Eventually, even if the brain is short, it's definitely of a bigger mass than the spinal cord. And so a big portion of the neurotube forms the brain, cranial 2/3. Then the caudal 1/3 forms the spinal cord. From the previous lecture on spinal cord development, you noted also that even though the neurotube comes from both primary and secondary neurulation is only responsible for forming the caudal part of the spinal cord, and perhaps from the sacral region going down. That is the only part of the spinal cord that is of secondary neurotube. Otherwise, the lumbar cord, the thoracic cord, and the cervical cord are all from primary neurotube. That is to tell you something, that the whole brain is from primary neurotube. And so even though neurulation was both from primary and secondary neurulation, as with regard to development of the brain, we'll just be focusing more about the primary neurotube and will not be talking about the secondary neurotube. So perhaps that's the last time I'm talking about the secondary neurotube. Subsequently, we'll just be talking about the neurotube, but that neurotube, you remember, it's actually the primary neurotube. All right. So that is the dorsal induction. And we've seen also agenda 2 there, parts and derivatives of the neurotube. Now I want us to talk about the second phase of development of the central nervous system, which is the phase of ventral induction. The phase of ventral induction largely occurs from the 4th to the 5th weeks of development. So you notice that neurulation, primary neurulation is already complete. And so ventral induction can begin even if secondary neurulation has not yet started. Ventral induction referred to the induction of the rostral part of the neurotube, which is the primary neurotube, so that it can differentiate to various brain vesicles. Basically, what happened during ventral induction is that the neurotube develops some swellings. These swellings are known as the brain vesicles. The process of ventral induction is largely directed towards patterning the forebrain. Remember, the forebrain is the cerebrum. It's largely directed towards patterning the forebrain. So we want to see how the cerebral hemispheres are patterned, how the encephalon structures come out, and the associated ventricular spaces, because that's basically the forebrain. However, even though it is largely directed towards patterning the forebrain, we will see that we will still have midbrain and hindbrain structures still arising from the brain vesicles as well. But the primary goal of ventral induction is to pattern the forebrain. And I want you to have that in mind, because when we're going to look at now disorders of ventral induction, we'll be seeing that they are basically disorders of cleavage, where there is failure of separation, perhaps, between the right and the left hemisphere and the like. Okay, so as we discuss the phase of ventral induction, we should now talk about the primary and secondary brain vesicles. Be reminded by this image first. So I've already shown you the first one there. That's a primary neurotube. But I want you to note this one. That the primary neurotube has a wall and a lumen. The wall is that one, this is the lumen. In the spinal cord, we saw how this happened. The wall has several cells which divide into some zones, giving rise to the different parts of the spinal cord in cross-section. The lumen becomes the central canal of the spinal cord. From primary neurulation, we know that we also have neurocrest, and neurocrest gives us various structures that we talked about. So when ventral induction occurs, so that the rostral neurotube is induced to form some swellings, how many swellings form? The first induction leads to the formation of 3 swellings. And these 3 swellings are known as the primary brain vesicles. We call them forebrain swelling, midbrain swelling, and hindbrain swelling. Well, there are better terms for them. We have what we call prosencephalon. And so prosencephalic swelling is the forebrain swelling. Mesencephalic swelling is the midbrain swelling. And rhombencephalic swelling is the hindbrain swelling. Note that each swelling has a wall and a cavity. That will be important, because the neurotube has a wall and a cavity. So even here, the swellings, we have a wall and a cavity. Remember when the cranial closed, so we will expect that to be blind as it is right now. You remember that the cranial 2/3 of the neurotube is the one that gives you the brain. So that's why even in this image, a bigger portion of the neurotube is shown to be giving the brain, and a smaller portion of it is the one giving the spinal cord, the caudal end of it, caudal 1/3 gives you the spinal cord. So these are the three swellings: forebrain, midbrain, and hindbrain swelling, or this one, forebrain, midbrain, and hindbrain swelling. Those three swellings are the ones that will eventually give us the brain. The spinal cord will come from the caudal 1/3 of the neurotube. Of course, the spinal cord will also come from secondary neurotube as well. So remember the three swellings: prosencephalon, mesencephalon, and rhombencephalon. From these three swellings, as development continues, we develop 5 swellings. Those 5 swellings are now known as secondary brain vesicles. Now, you're wondering in your mind, so you're coming from 3, how do you get 5? It would have been easier to say you get 6. Now, let's see how we get the 5 secondary brain vesicles from the 3 primary brain vesicles. The prosencephalic swelling, which is the forebrain swelling, differentiates into 2 types of swellings. There are some paired swellings which go right and left, so they are paired, large, spanning out. We call them telencephalic swellings, or you can just call it telencephalon. So these are right and a left telencephalon. And then another thing that also comes from prosencephalon is this swelling here, which we call diencephalon. So prosencephalon, which is a primary swelling, gives us 2 secondary swellings, telencephalon and diencephalon. Then mesencephalon, which is a primary swelling, does not change. So when you talk of secondary swellings, we also still have mesencephalon. It remains constant. So mesencephalon is both a primary swelling as well as a secondary swelling. How about rhombencephalon? Rhombencephalon gives us 2 swellings in a unique way. The 2 swellings of rhombencephalon are not completely separated in their cavity. So the cavity is just one large continuum, but from the wall, we see some separation. So the upper part of rhombencephalon becomes what we call metencephalon, and the lower part of rhombencephalon becomes the myelencephalon. So metencephalon and myelencephalon are secondary brain swellings, which are derived from rhombencephalon, which is a primary brain swelling. So what do you notice? In total, we have 5 secondary brain vesicles, yet we had 3 primary brain vesicles. Remember still, each vesicle, whether primary or secondary, has a wall and a cavity. As the brain vesicles form, there's some bends that also occur on the brain, and these bends are called the brain flexures. We have primary and secondary brain flexures. A primary brain flexure is an anterior or a ventral flexion, ventroflexion. So the neurotube bends ventrally, and a secondary brain flexure is a dorsal flexion. So the neurotube flexes on the dorsal side. Having said so, let's look at this. Imagine this was a straight thing, but now it has already bent on 2 ends. because these ones are ventral flexures, ventral flexions, they are primary brain swelling. There's one that form at the level of the mesencephalon. We call it the cephalic flexure. So it makes the forebrain to tilt ventrally, as you can see there. It occurs at the level of the mesencephalon, so sometimes it may be called the mesencephalic swelling. Then we have this other one that forms between rhombencephalon and the future spinal cord. We call it the cervical flexure. Cephalic flexure and cervical flexures are primary brain flexures. They are ventral flexions. They make the brain to tilt twice, ventral side. So that will make the brain to bend so much. Now, to ensure that now the forebrain comes up, we then need a secondary flexure, which is a flexion on the dorsal aspect, this one. So this one makes the brain to tilt backwards. Then that flexure that occurs on the dorsal aspect occurs at the level of pons, and so we call the pontine flexure. So take-home message is that pontine flexure is a secondary flexion, and it's on the dorsal aspect of pons, while cephalic and cervical flexures are primary flexures. They are ventral flexions occurring at the level of mesencephalon and the junction between the brainstem and spinal cord. We can appreciate that on this other image as well. So this is the level of the cervical flexure. This is the level of the cephalic flexure. And that's the level of the pontine flexure. Now that we've talked about the primary and secondary brain vesicles and a ventral induction, let's see what these vesicles will eventually become. What does the telencephalon become, the encephalon become, and the like? We want to see the derivatives of the vesicles. Remember, we are still talking about ventral induction, yes, that the brain has been induced to form vesicles. But we want to see eventually what do these vesicles become. So as we discuss this, I want you to know that the wall of a vesicle will give us something, but the cavity of a vesicle also gives us something. So as we discuss the derivatives of the vesicle, we have to discuss the derivatives of the wall as well as the cavity of each vesicle. This image captures much of this story. So the first image shows us the three primary brain vesicles: prosencephalon, mesencephalon, and rhombencephalon. The prosencephalon gives us two vesicles, telencephalon and dencephalon. Mesencephalon remains constant. Rhombencephalon gives us two swellings, metencephalon and melencephalon. And so these five are secondary brain vesicles. They each have a wall and a cavity. Now let's see, what do these vesicles become? We'll start with telencephalon. Telencephalon gives rise to the cerebral hemisphere. Now, the wall of the telencephalon will give us the tissue of the cerebral hemisphere, while the cavity of telencephalon will give us the lateral ventricles. Remember, the lateral ventricles are within the hemisphere anyway. So because we have right and left lateral ventricle, we also have a right and left cerebral hemisphere. And we also have a right and left telencephalic vesicle. When you talk of the wall of telencephalon giving you cerebral hemisphere, I want you to understand that that will involve the cerebral cortex, the white matter of the cerebrum, and even the basal ganglia. Structures of the limbic system are also there. So don't just think it's the cortex only. It's the cortex, yes, but we also have the white matter. We also have the basal ganglia, and limbic structures are also there. Those are telencephalic derivatives. The cavity gives you the lateral ventricle with all its parts, the anterior horn, the body, the posterior horn, the inferior horn, as well as the antrum of the lateral ventricle. They all come from the cavity of telencephalon. How about dencephalon? Dencephalon gives us basically the components of the dencephalon that we know from gross anatomy. So the wall of dencephalon gives you the thalamus, hypothalamus, subthalamus, and epithalamus. So those ones come from the wall of dencephalon. While the cavity of the dencephalon gives rise to the third ventricle. That's why we see this continuity here from the cavities, which are future lateral ventricles. We can see that continuation to the cavity of the third ventricle. So I want you therefore to imagine that this point will come like this as a narrowing. So there'll be a narrow channel between this space and that space, and that is what we'll call the interventricular foramen of Monro on one side and also on the other side. So the connection between the lateral ventricular space and the third ventricular space will be a narrow channel, interventricular foramen of Monro. When these vesicles will have undergone complete separation or near complete separation, as we know from gross anatomy. Now let's come back here. I've told you that the wall of the dencephalon gives you thalamus, hypothalamus, subthalamus, and epithalamus. That's true. But there are other things that may also come from dencephalon. For example, the floor of the dencephalon is the one that will give us the downgrowth that forms the posterior pituitary gland. So the floor of the dencephalon gives rise to the posterior pituitary gland, what we call neurohypophysis. Remember, the pituitary gland has two parts, anterior and posterior. The anterior pituitary usually is a derivative from below. It's a structure from below called the Rathke's pouch that will develop from the roof of the mouth going up. But posterior pituitary grows from the floor of the dencephalon downwards, and then the two shake hands somewhere. The other thing that may be arising a bit earlier, and so maybe I'd prefer to say it arises from the wall of prosencephalon, rather than limiting ourselves to these two, is what we call the optic vesicle. The optic vesicle is the embryonic structure that comes from the wall of prosencephalon and grows out to give rise to the retina and the optic nerve. So the retina and the optic nerve is also a derivative of the wall of prosencephalon, although extrapolated here, many books will also show that perhaps it will be attaching to the dencephalon, or sometimes some books show it attaching to the telencephalon. But the concept here is that that thing comes a bit earlier, before even the separation of the secondary vesicles. But eventually, yes, it was part of this. Remember, the retina and the optic nerve, they are just extensions of the primary neurotube. And that's why some people say the retina and the optic nerve are actually part of the central nervous system and maybe not peripheral. And now we understand why they say so. Okay, how about the third secondary vesicle, mesencephalon? The wall of mesencephalon gives rise to the midbrain, while the cavity of mesencephalon gives rise to the cerebral aqueduct of Sylvia, the one that connects the third to the fourth ventricle. Back to that midbrain concept, usually the wall of mesencephalon will be similar to what we saw, or near similar to what we saw with the wall of the developing spinal cord, where we had alar plates and basal plates, which are swellings of the mantle layer of the neurotube. And remember, alar plate gives a sensory nuclei, and that also happens even in the midbrain. And basal plate gives rise to the motor nuclei, that also happens in the midbrain. So midbrain and spinal cord share a lot of similarity in terms of development. The only unique thing is that now maybe midbrain will not have those many nerves coming out or entering from neurocrest. Right, that's midbrain. How about metencephalon and myelencephalon, which are derivatives of rhombencephalon? The wall of metencephalon gives rise to pons and cerebellum, while the wall of myelencephalon gives rise to the medulla oblongata. However, the cavity of rhombencephalon doesn't really segment much, and so it remains as one large cavity, and that is the fourth ventricle in actual sense. So the cavity of rhombencephalon, the cavity of hindbrain swelling, is the fourth ventricle. So if you wanted to be more politically correct, then you say the cavity of metencephalon gives you the upper part of the fourth ventricle, and the cavity of myelencephalon gives you the lower part of the fourth ventricle. But as you know from gross anatomy, the fourth ventricle is just a rhomboid structure, a large cavity really, not segmented. And so just understand that concept as well. The cavity of the hindbrain swelling gives you the fourth ventricle. So these are the derivatives of the secondary brain vesicles. Now we've done that aspect. The image here captures that as well, also putting into perspective the flexures of the brain that have already occurred. In particular, you also capture that there's some things I told you about the optic nerve, as well as perhaps the ones about the pituitary that we also see coming out. Let's now talk about the third phase of development of the brain. That is the phase of neuronal proliferation and migration. This phase may be a bit tricky, a bit technical, but I'll try to keep it as simple as I can so that we're on the same page. The phase of neuronal proliferation and migration is primarily aimed at development of the cerebral cortex. This phase involves 4 key events, and let's understand these 4 key events. One of the events that take place is neuronal proliferation, which means that we are having numerical increase in the number of neuroblasts. This occurs through mitosis. And as a matter of fact, neurogenesis occurs in the brain beyond what is required. So the number of neurons that form during embryonic development are way more than what the brain actually will ever need. The neurons are produced in excess. Why they have to be produced in excess, maybe we may not have the full wisdom to understand. But yes, they're produced in excess, and eventually some of them will die actually. So neuronal proliferation is the first aim, just produce the number of neurons, to add to the numbers of neuroblasts. Apart from neuronal proliferation, the neurons also migrate. Once they've been formed, they move. And the movement of neurons is largely from deeper zones to superficial zones of the neurotube. If you remember the wall of the neurotube, maybe I'll be showing you again, but remember the wall of the neurotube has inner zones, the ventricular zone, going outwards to the superficial zones. Neuronal migration occurs in that direction largely, from deeper to superficial zones. Remember, the major aim of neuronal migration is to develop the cerebral cortex. And where is the cerebral cortex? Superficially. The other event that characterizes neuronal proliferation migration is basically differentiation and cortical organization. This is in the understanding that we are aiming at forming the cerebral cortex. So the neurons which go to the cerebral cortex differentiate. Some of them become the pyramidal neurons. Some of them become the granule cells. So they differentiate. Other than differentiation, there is cortical organization. And what do I mean by cortical organization? The neurons organize themselves in the cortex so that eventually we also have gyrification and sulcation. So when I'm talking about cortical organization, I'm also talking about how the gyri and sulci also come to be in the cerebral cortex. Lastly, we have programmed cell death of the neurons. Understand that the neurons that formed were more than what is required. And so some of them have to die really. Neuronal death is also part of development of the central nervous system. It's part and parcel of the development of the cerebral cortex. Many neurons die. So those are the 4 key events that characterize cortical development largely. Maybe just to put more meat to that story that you said, I'll first explain to you or remind you about the cross-sectional layout of the neurotube so that we understand how neuronal migration is actually occurring. Look at the first image. That is how the neurotube looks like, the primary neurotube, remember? That's how it will look like, the wall and the cavity. And at the level of the spinal cord, this is what we saw, that yes, the wall will proliferate eventually, so that you'll have a cavity that's thinning out, will come the central canal of the cord. This wall divides into 3 zones: ventricular, intermediate, and marginal zone. The intermediate zone, in this case, will give us the gray matter, and which will be the one dividing into basal plate and alar plate. The marginal zone is here, which in the spinal cord contain axons of neurons, or basically white matter. However, we know that the story is a bit different in the brain. You know, in the brain, the cortex, gray matter is outside and white matter is inside. In the spinal cord, the white matter is outside, the gray matter is inside. So we will expect to make the gray matter be outside on the brain, in as much as in the spinal cord, the gray matter is inside. So that is why neurons must migrate from deeper zones to superficial zones to form the gray matter in the brain. Look at this. So this is a neurotube. Well, maybe it's almost completing the fold, but that's the wall of the neurotube. There's very early cellular differentiation in the wall of the neurotube. The cells differentiate in the wall of the neurotube quite early. These cells differentiate into two lineages largely. The ones designated to become neurons are called neuroblasts. The ones designated to become supporting cells are called glioblasts. So we have very early cellular differentiations, neuroblasts and glioblasts differentiating quite early. When you look at the epithelium, it appears to start for the columnar epithelium to mean that, well, maybe all the cells are still resting on the basal lamina, but we have a mixture of short and tall cells. But as time goes by, we'll have true stratification of this epithelium anyway. I've already told you the names of the earliest cells that will be there. Neuroblasts are the stem cells for neurons, and the glioblasts are the stem cells for the supporting cells. In this case, the glioblasts are the ones that will give rise to ependymal cells. They'll give rise to astrocytes. They'll give rise to the oligodendrocytes because this is CNS. They don't quite give rise to microglial cells. Those ones arise from mesenchyme because they're basically connective tissue cells, they're macrophages. Remember that the wall of the neurotube differentiates into three zones. The innermost zone is called the ependymal zone. The middle zone, intermediate zone, is called the mantle zone. And the outermost layer is called the marginal zone. This is what we saw when we looked at development of the spinal cord. Back to here, the ependymal zone or the ventricular zone, as we saw, is a region that contains the stem cells. Both glioblasts and neuroblasts are there. And so it's a zone of active mitotic division. Having said so, cells will be arising from that zone, going peripherally. And so if you're going to develop the cerebral cortex, cells will be migrating from this zone. Because of its origin in giving rise, a rich origin of cells that eventually migrate to form the cerebral cortex. In the brain, we prefer calling this one the germinal zone or the germinal matrix. And so those are some of the terminologies that may be used. It shows it is a region that contains population of stem cells, the germinal matrix or the germinal layer. Quite important is that some glial cells differentiate quite early as well, so that they can guide the migration of the neurons. The name given to the glial cells that might differentiate early are radial glial cells. These radial glial cells differentiate quite early and they form a scaffold where now the neurons will use to move away from the germinal matrix. Look at this image. So those lines you see are representing the radioglial cells. Neurons will be proliferating from this germinal matrix and moving outwards. So this scaffold will be guiding the migration of the neurons away from the germinal matrix towards the cortical plate. The role of the radioglial cells is to provide a scaffold for neuronal migration. Significant neurogenesis occur during the 10th to the 20th week of development. That's when most neurogenesis take place. The cells that proliferate will be migrating, as we said, from the ventricular zone peripherally. The migration path is in multiple waves. So there are multiple waves of migration from the ventricular zone towards the periphery. And I told you that that region that is giving you the stem cells is what you call the germinal matrix. I want you to capture that there are multiple waves of migration. And those multiple waves of migration is what will eventually lead to the formation of a multi-layered cerebral cortex, because there are multiple waves of migration. Usually, the later waves form more superficial layers. The earlier waves form the deeper layers of the cerebral cortex. All right, there are two patterns of neuronal migration. We have what we call the radial migration, which is what we've largely talked about. This is where neurons migrate along the path of radioglial cells. So just direct from ventricular surfaces to the cortical surface. But there's another trajectory path, which we call tangential migration. This is migration along the transverse surfaces of the neurotube, or the developing brain, basically. Maybe if I use an image that will help us understand those two concepts. Consider this to be the wall of the neurotube. From the deeper surface to the superficial zones. So germinal zone is this one here, that is giving rise to the neuroblast that are supposed to migrate. So this is ventricular space, and perhaps that the pile surface. So ML here refers to the molecular layer of the cerebral cortex, and CP here refers to the cortical plate, where we want to form the other layers of the cerebral cortex, cortical plate. IZ here means the intermediate zone, the zone that is going to be the future white matter. Because remember, in the brain, the cortex, gray matter is outside and white matter is inside. GE here means ganglionic eminence, and this basically represents the nuclei within the cerebrum, maybe thalamus or basal ganglia. So this nuclei within the cerebrum, that's ganglionic eminence. So let's understand the trajectory paths. The one labeled 1 shows us a migratory path from the ventricular zone or the germinal zone towards the periphery. And we see that it's following the scaffold of the radioglia. So that is the radial migration trajectory. That's the trajectory of radial migration, the one labeled 1. Compare that with the one labeled 2, which is now showing us tangential trajectory pattern. That some neurons migrate in a transverse path along the transverse surfaces of the neurotube wall. So that's tangential migration. But what do you notice? Most of these neurons that have tangential migration eventually then also have radial migration paths. So they are coming from the germinal zone, they might be migrating that way, that eventually they migrate that way. About 3. We can see that 3 is a migration path that is almost transverse. So it's also a tangential migration. Within the intermediate zone is a prospective white matter. And these neurons are originating from ganglionic eminence, which I told you would be perhaps representing the thalamus or basal ganglia. So the neurons from there also migrate tangentially. CP is a cortical plate and ML is the molecular layer. The cortical plate represents the layers of the cerebral cortex, maybe apart from the molecular layer. So remember, within here, we will have then, if that is molecular layer, then within here we'll have the outer granular layer, outer pyramidal layer, inner granular layer, inner pyramidal layer, and the multiform layer. The cortical plate represents the region that will be becoming the cerebral cortex. The neurons that migrate, the earlier neurons that migrate will occupy these deeper zones, and the later waves of migration occupy more superficial zones. We've said many things about neuronal migration, but let's just bring some things into perspective. The neuronal migration process varies for each region. So how they migrate in the cerebellum may be different from how they migrate in the cerebrum, and how they migrate in the frontal cortex might be different from how they migrate in the insular lobe. However, there's some principles that we can take home. The ultimate migration path is from deeper zones to superficial zones of the neurotube. That's the ultimate. So whether it was taking a radial trajectory pattern or a tangential trajectory pattern, ultimately neurons migrate from deeper zones to superficial zones. The other take home is this, that we have multiple migratory waves, and these multiple migratory waves are the ones that lead to the formation of a multi-layered cortex, because there are multiple waves of migration. Within the cerebral cortex itself, neurons which are of a later wave will pass through preceding layers to form more superficial layers in the cortical plate. So later waves of migration form more superficial layers of the cerebral cortex. The slide here tries to capture what I've told you about later waves forming more superficial zones. Let's start from A, which basically shows us the wall of the neurotube with the piled surface and ventricular space there. So that's the pseudostratified, perhaps columnar epithelium of the neurotube. Then the one labeled B is showing us some differentiation of the wall. So we have the ventricular zone already established. And then now we have the marginal zone also established in this particular second image. In the third image, we now have all the three zones established. Marginal zone, intermediate zone, and the ventricular zone established. So this is what we're talking about when we say that the wall of the neurotube has three layers from inside to outside: ventricular zone, intermediate zone, and marginal zone. In the brain, the marginal zone, in the brain, the intermediate zone will give us the white matter and not the gray matter. Remember, in the spinal cord, what is intermediate here will be the mantle layer, which will give rise to the gray matter. But in the brain, the intermediate zone become the white matter eventually, and we can follow that to that end. It becomes the white matter. The marginal zone in the spinal cord became the white matter, but in the brain, it forms the molecular layer of the cerebral cortex. So the marginal zone becomes the molecular layer of the cerebral cortex, and that is why we can follow that up to that end. Now, as neurons migrate in waves, so in this D image, we see a layer that is now appearing called the cortical plate. As waves of migration continue, we see that that layer continue to become bigger. And so we see there, the cortical plate now represents the other layers of the cerebral cortex. And as I told you, later waves of migration occupy more superficial zones of the cortical plate. Right, the layer of stem cells is basically that particular region, that subventricular zone, which contain the layer of stem cells that are continue to migrate. All right. Now this happens as development continues. The gyri continue to form as the cortical development also continues. Look at these images. The first one at 25 weeks, look at the level of gyrification and sulcation. Yes, it has started, but not complete. Cleavage has already occurred, as we can see. So the right and the left telencephalic vesicles have already been cleaved, as we can see, but not completely do we have the sulcation. Maybe just one sulcus has formed there. As time goes by, we can see the number of gyri and sulci are increasing. And at maturity, where maturity means term here, we now see that we have complete gyrification and sulcation. So the gyrification and sulcation become as a result of migration of neurons and also cortical organization. Great, so that's the story of development of the brain, basically. But let's talk about now the phase of neuronal myelination. This is the fourth phase, neuronal myelination. Neuronal myelination in the central nervous system begin around the 5th month of fetal life and basically continues throughout life. However, the myelination of embryonic development of the brain occurs in a defined order. Yes, it will begin during the fetal period and usually also extend up to the first month of extra-uterine life. In your brain, myelination still happens, but that's not necessarily myelination of development. So I'm talking about the myelination of normal development. That one occurs during fetal life and the first year of life. Predominantly, that's when it occurs. Thereafter, very minimal, but of course, you can still have myelination for repair. Generally, myelination in the brain proceeds in a particular pattern from caudal to rostral and from posterior to anterior. So from caudal to rostral and from posterior to anterior. That's the general direction of migration, of myelination of neurons. This slide is not really for remembering actively, but just highlighting for us that concept that not all neurons are myelinated at the same time. Importantly, I want to capture that the frontal lobe takes it last actually, and that the peripheral parts of the frontal lobe are myelinated later than the central parts of the frontal lobe. Occipital will be myelinated a bit earlier than frontal lobe. Again, even in the occipital, the central part will myelinate earlier than the peripheral parts. Okay. Just to finish the story. So this is how the mature human brain look like in MRI. The images shown here are MRI images. The first one is sagittal, the second one is axial, and the third one is coronal. There are different MRI imaging modalities that we use in studying the brain. The one we are seeing here shows us white matter as white and appearing brighter than the cortex, which is the gray matter. When we see white matter as white and gray matter as gray, then we call that one T1 weighted MRI. And so these are T1 MRI images on the sagittal, axial, and coronal section. In T1 MRI, fluid spaces will be dark, as we can see in this image. So these are the lateral ventricles. So this white one is the splenium of the corpus callosum, and that's the genu of corpus callosum with the forceps minor and forceps major, that one. Got this with the basal ganglia, and that these are the thalami. There's anterior horn of the lateral ventricle, and that's posterior horn of the lateral ventricle. Looking at this one, that's lateral ventricle. This is fourth ventricle. So these are lateral ventricles as well. In this second one, what do we notice about the color of white matter and the cortex? The white matter is darker than the cortex. When we see white matter darker than the cortex, we call that T2 MRI. In T2 MRI, white matter is dark, the cortex is lighter, and in particular, fluid is bright, as we can see. So these are T2 MRI images. We also use these ones to study the brain. How about these ones? These ones, the white matter, they're all axial intentionally. The white matter is darker than the gray matter, so that's still T2. It's a special type of T2 because here the fluid is not bright as we expected to see in T2. Here the fluid is dark. We call this one T2 flare. Flare stands for fluid attenuated inversion recovery. You don't have to remember that jargon, just say T2 flare. We use T2 flare largely in studying pathologies of the brain. We don't have to go into the details of that, but yes, you may sometimes see the brain in T2, and basically that's still white matter. This is the region of the basal ganglia, that's thalamus as well. These are the eyes. You see now the fluid in the eye is also dark in this particular image because the fluid has been nulled. How about these ones? This one, the unique thing we are seeing, we can't really differentiate gray and white matter nicely. But yeah, we can still pick it. The white matter is darker, the gray matter is lighter. And importantly, also the fluid spaces are dark. But the most shouting thing in this image is that the bone is very bright. When you see bone very bright, that is CT scan. So this is axial CT, this is coronal CT, and this is sagittal CT of the brain. This is CT scan. We also use CT scan sometimes to study the brain for other reasons. Great, so we will stop there. We've looked at development of the brain, and we've looked at the 4 phases of development: dorsal induction, ventral induction, neuronal proliferation and migration, and neuronal myelination. But as we did those 4 phases, we've also looked at the brain, we looked at the neurotube and its derivatives. We also looked at the brain vesicles, primary and secondary brain vesicles, and what each vesicle become. And we also looked at the flexures of the brain. Thank you very much. The next neurodevelopment lecture, which will be the last one, will be on congenital malformations of the brain and the ventricular system. So you can watch out for that. Thank you very much. We'll stop there.